In this video, we will examine the process known as translation. Translation is the process by which proteins are synthesized by ribosomes as directed by the message within the mRNA. Before we begin to examine the events in the process of translation, let's first examine how the linear sequence of nucleotides of the mRNA can be translated into a linear sequence of amino acids. The ability to convert DNA to RNA is relatively easy to understand because a newly synthesized RNA sequence is complementary to that of the DNA strand it is synthesized from. The conversion of RNA to amino acids is not as easily understood. RNA is composed of 4 different nucleotides, and amino acids exist as 20 different types in most cells. Therefore, a one-to-one correspondence between a nucleotide and an amino acid would not work. Even if 2 nucleotides were used in combinations, only 16 different amino acids could be coded for. Scientists found that the mRNA is read consecutively in sets of 3 nucleotides. RNA read as triplets allows for 64 possible combinations of the 4 nitrogenous bases. The sets of 3 nucleotides on the mRNA are known as codons. The 64 different possibilities allow for some of the amino acids to be coded for by more than one codon, and 3 of the codons to serve as stop codons for the process of translation. The rule specifying the correspondence between a nucleotide triplet and the amino acid of a protein is known as the genetic code. 2 important characteristics to take note of in regards to the genetic code are its redundancy and the universal nature of the code. When we speak of the redundancy of the code, we are referring to the fact that more than one codon can code for the same amino acid. The redundancy of the genetic code protects against detrimental mutations by having more than one codon coding for the same amino acid. The universal nature of the genetic code means that almost all organisms use the same genetic code to produce their proteins. There are few exceptions to the code seen in the image. Those exceptions primarily occur in the mitochondria, in bacteria, and in some fungi and protozoans. Translation requires several forms of RNA as participants to complete the process. Let's examine the role of messenger RNA, transfer RNA, ribosomes, and ribosomal RNA in the process of translation. Messenger RNA molecules are the form of RNA that carries the genetic information for the order of the amino acids of the protein to be synthesized. Prokaryotic and eukaryotic mRNA molecules differ. Prokaryotic mRNA molecules contain the sequence for several genes and therefore code for more than one protein. Eukaryotic mRNA molecules contain the nucleotide sequence of only one gene and therefore contain the code for a single protein. In addition to coding for a single protein, the eukaryotic mRNA must be processed to convert pre-mRNA to a functional mRNA. The events needed to process eukaryotic mRNA are discussed in the lecture video, Gene Function Part 1. Another major difference between bacteria and eukaryotes is the location in which the mRNA is utilized to produce proteins. Prokaryotes can carry out transcription and translation simultaneously because the prokaryotic mRNA is transcribed in the cytoplasm and does not require additional processing before being used in translation. Eukaryotic mRNA, however, is made in the nucleus and must undergo processing before leaving the nucleus. The eukaryotic mRNA then travels to the cytoplasm to be translated by ribosomes. Transfer RNA molecules are important forms of RNA that are needed to transfer the correct amino acids to the ribosome for translation. Transfer RNA is a single-strand RNA molecule that folds back on itself, forming hairpin loops. The bottom hairpin loop contains a 3-nucleotide sequence known as an anticodon that is complementary to a codon on the mRNA. On the 3' end of the tRNA molecule, an acceptor stem is present that is designed to carry a specific amino acid. Enzymes present in the cytoplasm add the appropriate amino acid to the acceptor stem, a process referred to as charging the tRNA. Some of the tRNA molecules that exist within a cell are able to recognize more than one codon. Such tRNA molecules require accurate base pairing with the mRNA codon at only the first two nucleotides and tolerates a mismatched pair at the third nucleotide. This phenomenon is referred to as wobble base pairing and allows a cell to match the amino acids to their appropriate codons with fewer tRNA molecules. The number of tRNA molecules varies depending on the species. For example, E. coli uses approximately 40 different tRNA molecules to match all 61 codons, while humans use 48 different tRNA molecules to match the same 61 codons. Ribosomes are the cellular structures which serve as the sites of translation within cells. As we saw in Unit 1, ribosomes are comprised of proteins and ribosomal RNA. Prokaryotic and eukaryotic ribosomes differ in the types of proteins and rRNA that make them up. Prokaryotic ribosomes are described as 70S ribosomes that are made up of a large 50S subunit and a small 30S subunit. Eukaryotic ribosomes located within the cytoplasm and in association with the rough endoplasmic reticulum are 80S ribosomes that consist of a 60S large subunit and a 40S small subunit. The larger size of the eukaryotic ribosomes reflects the fact that they contain larger rRNA molecules and more protein than those of the bacterial ribosomes. The ribosomes of the eukaryotic mitochondria are 70S ribosomes and are similar to those of bacterial cells. We will see in Unit 4 that these structural differences allow us to utilize drugs that target 70S ribosomes to selectively kill bacteria. We will discuss such drugs in more detail in Unit 4. The small subunits of ribosomes are built to accommodate 3 codons at the same time. Each of the sites that accommodate a codon is named after the function of the site. Let's look at the sites from left to right on the small subunit seen in the image. The first site seen on the far left of the small subunit is called the E site or exit site. The E site is the location at which discharged tRNA molecules move through to move out of the ribosome. The middle site is the P site and is named so because it holds the tRNA molecule that is bound to the growing polypeptide chain. The last site, the A site, is the site in which arriving charged tRNA molecules enter the ribosome to deliver their amino acids. Now we will examine the specific events of translation. Translation occurs in 3 stages: initiation, elongation, and termination, each requiring protein factors and energy. We will examine the steps as they occur in bacteria first and then compare them with eukaryotic translation. Initiation results in the formation of an initiation complex. The first event that must occur to form this complex is to bind the smaller subunit with the mRNA transcript at the appropriate location on the mRNA. For a bacterial mRNA transcript, the region that acts as the ribosome binding site is known as a Shine-Delgarno sequence. When the small subunit is bound to the ribosome binding site, it positions the start codon, AUG, in the P site of the small subunit. A transfer RNA molecule carrying the amino acid formylmethionine with an anticodon complementary to the start codon will be bound in place as a molecule of GTP is spent. This is then followed by the binding of the larger ribosomal subunit to form an initiation complex. Once the initiation complex is formed, elongation of the polypeptide can proceed. The next transfer RNA molecule with an anticodon complementary to the codon will move into the A site. In the image, the second codon is UUU. Therefore, the appropriate tRNA will have the anticodon AAA. This tRNA will carry the amino acid phenylalanine. Energy is required for the tRNA to bind to the A site. After binding to the A site, a portion of the large subunit of the ribosome acts as an enzyme to catalyze the formation of a peptide bond between the amino acid in the P site and the one held in the A site. The polypeptide chain remains attached to the tRNA in the A site. More energy in the form of GTP is then used to shift the ribosome down the mRNA molecule one codon. This shift causes the empty, uncharged tRNA to move into the E site, the tRNA with the polypeptide chain to move to the P site, and opens up the A site to receive a new charged amino acid. The uncharged tRNA would be released into the cytoplasm so it could again be charged. These steps would continue to cycle until the ribosome reaches a stop codon. Once the ribosome has reached a stop codon, a portion of the large subunit, along with proteins known as release factors, sever the polypeptide from the last tRNA. The ribosome then dissociates into its two subunits and releases the mRNA transcript. Multiple ribosomes may translate the same mRNA simultaneously. As one ribosome begins to elongate a polypeptide, another ribosome may bind to the same ribosome binding site immediately behind the first one to begin translation. This could continue with one ribosome after another associated with the same mRNA, allowing many polypeptides to be produced from a single transcript. The order of events of translation in bacteria is similar in eukaryotes. There are some important differences to take note of that exist between the two. First, the initiation of translation occurs when the small ribosomal subunit binds to the 5' guanine cap of the mRNA rather than a specific sequence. A second key difference is the first amino acid coded for is methionine rather than formylmethionine. Eukaryotes are also capable of producing proteins using ribosomes that assemble with the rough endoplasmic reticulum, allowing them to package proteins for export.